Everyone, I am going to explain DNA microarray basics and application. This is the outline I am going to explain. Genetics versus genomics, basics of gene expression, what is the types, procedure and applications of DNA microarray. We all know what is the genetics is the study of heredity. With the help of genetics, we are able to know the likelihood of developing disease, and we are able to detect the mutation but not the disease. On the other hand, genomics is the study of genes, their interactions and their expression. With the help of genomics, we are able to know how aggressive is the cancer, that is the prognosis, and what is the likely benefits from the treatment, whether the treatment is effective or not effective for particular tumor or disease. So human genomic project started from 1984 and completed in 2003. The key features of this project is there is a 20,500 genes in our body. So it is very interesting to know the expression of that gene, because not 20,000 genes are activated at particular time, they are activated simultaneously. So how the 20,000 genes, 20,500 genes are expressed in our body, this is the central dogma of life, from DNA to RNA and RNA to protein. So suppose after eating food we need insulin. Genes in our pancreas get activated that send messenger RNA. Messenger RNA is nothing but the code of sequence for protein synthesis, and final protein is insulin. So in this way, after eating food, our gene for insulin synthesis are activated and rest of the time they are inactivated. So with the help of this is the very basic example. With this explanation, we are able to know that not all the 20,500 genes are activated at particular time. They are activated whenever they are required. Okay, so what is the gene? We should know the basic concept before understanding the DNA microarray. Genes is nothing but the part of DNA. Okay, it is the nucleotide sequence. There is a 4 nucleotides A, T, C, G. This is the nucleotide sequence on the DNA. One DNA containing many genes. Some are too small, some are too large. It is nothing but the part of sequence. So DNA, with the help of histone protein, again condensed to form nucleosomes. Nucleosomes again condensed to form chromatin, and chromatin again condensed to form the chromosomes. So ultimately, chromosome is nothing but the condensation of DNA. Cells containing 46 chromosomes, and that 46 chromosomes containing 20,500 genes. Okay, so each cells of our body containing 20,500 genes. So this is the sequence of nitrogen base, that is C always bind with the G, A always bind with the T. ATGC, it is the sequence on the DNA. It is called the gene. So human gene is not whole DNA is not full of information. It is divided into two parts, that is exon and intron. So intron part is not having any information. The informative part of DNA is exon, which is so intron part is removed by DNA splicing. So in this way the messenger RNA is formed. Messenger RNA is nothing but the code of information. So this is the basic example of regulatory and structural gene. This is the example of muscle cells. Okay, so in our muscles there is a gene for actin synthesis and myosin synthesis. So that send messenger RNA, and final result is formation of protein, that is actin and myosin. But our muscles containing melanin and insulin gene also, but our muscles can't able to synthesize insulin or melanin. Why? Because this is under control of this regulatory gene. Okay, so in this way the particular organ is responsible for secretion of particular protein. So the actin is myosin act as a structural gene, and the melanin and insulin off, which is under control of the regulatory gene. Okay, So this is the gene expression starting from the promoter region of the DNA, the 5s to 3, and there is a termination region. Okay, this is the basic sense concept. So on the DNA there is a formation of transcription bubble on template strand. So 5s to 3 there is a formation of mRNA. After formation of mRNA, it's moved toward the ribosomes for synthesis, protein synthesis. Okay, the ribosomes having a larger unit and one smaller unit. Ribosomes containing 3 parts, A, P and E. So on the smaller unit there is a messenger RNA, and on the larger unit the tRNA, that is clover leaf like arrangement, comes with one amino acid attached to on the head of tRNA. So tRNA containing anticodon, and the messenger RNA is having codon. So tRNA comes to the A site, move toward E sites and remove from the E sites. Now the next tRNA come toward the A sites. In this way there is a continuation formation of peptide chain. Peptide chain is nothing but the sequence of amino acid. There is the final product of protein synthesis. So until and unless there is a stop codon on the mRNA, the chain is continuously synthesized. So there is the 3 stop codon, that is the UAG, UAA and UGA. Okay, this is the 5s to 3. mRNA is attached on the lower sides and the tRNA come from the other sides, and there is a formation of the peptide chain, okay, on the head of tRNA. So in this way the final product is the protein synthesis. Okay, now why I am emphasizing so much on gene expression? Because the gene expression is very easiest to measure. Okay, there is a two way to detect the expression. Either we can detect the DNA expression or we can detect the protein expression, but the protein, which is the final product, is the too dynamic. They are very rapidly created and rapidly destroyed. That's why the messenger RNA or DNA synthesis is very easier to measure. So the unique pattern of gene expression for a given cell or tissue is referred to as molecular signature. Okay, now why the gene expression is important in malignancy or any disease? I am explaining with the help of this simple example. There are the 3 genes, gene A, gene B and gene C. Gene A is overexpressed in normal cells, gene B is not expressed, and gene C is underexpressed. This is the normal cells. Now come toward the cancer cells. Gene A, which is overexpressed in normal cells, are not expressed. Okay, not expressed in cancer cells. On the other hand, gene B, which is not expressed in normal cells, are overexpressed in cancer cells, while the expression of gene C is unchanged. So we can easily come to know with the point that gene B overexpression result in that particular cancer. Okay, this is the very basic example of DNA microarray. DNA microarray doing nothing but the measure the abnormal gene expression. So this is the DNA microarray machines and the printer. Historical background. Let's review some historical background. Every person on the screen having some contribution of DNA microarray. Like principle of DNA microarray is based on the southern blotting technique. Concept of DNA microarray began in the mid-18. The robotic system was developed by the Liarch group in 1919, and Steve Fowler developed the scanner for DNA microarray, and finally gene expression pattern with the complementary DNA microarray reported by the Patrick Brown and his colleagues, Mark Shena and his colleagues. Okay, so what is the meaning of array in the DNA microarray? Okay, so array is nothing but the small tiny holes on the DNA glass slides. Suppose this is the glass slide, this is the area of 1 by 1 centimeter, 1 by 1. It's having multiple tiny holes. We can use up to 20,500 holes for every representative gene. We know our body, human body is having 20,500 genes. So we can use 20,000 up to the 20,500 holes. So the every hole is having the complementary copy of DNA, which is produced by the PCR machine. Okay, so every hole containing complementary copy of DNA. Okay, this is the meaning of array. So we can use first either conventional glass slide, or second is the more advanced is the DNA microchip. Okay, DNA microchip, we are not using whole genes as we using whole genes in the glass slides, but in the DNA microarray chip we are using nucleotide, short nucleotide, that is the oligo means short and nucleotides means the sequence. So we are, for example, we know the gene 1, we know every sequence of 20,500 genes. So it's very easy to make the complementary copy of that particular gene or the gene of interest. Suppose gene number 1 is having A, T, C, G sequence. So we know the rule, A always bind with the T and C always bind with the G. So we make a complementary copy having sequence T, A, G, C. In this way we can put this complementary copy on hole number 1 on DNA chip. So this is the basic concept of DNA microarray. We can use either conventional glass slide or more advanced DNA chip. So the principle of DNA microarray is nothing, is only simple hybridization. So suppose this is a glass slide, this is having one hole containing DNA. Okay, suppose this is the DNA. We know we are using DNA gene which we want to measure the expression. That particular gene is known as the probe. Okay, then we mix the DNA of gene of patient plus control. We know the rule of complementary. The DNA which is more complementary to the DNA which is present on the glass slide will bind it to more firmly and give that particular color. That is the principle of DNA microarray. It is nothing but the hybridization. Okay, microarray is the snapshot of the which capture the activity of thousands of genes at particular time, which is in the medical term we are using gene expression. It's nothing but the captured activity of thousands of genes at particular time. So the basic question is why we need DNA microarray. So before invention of DNA microarray, we are not able to use the simultaneous activity of all 20,500 genes at particular time. We are only able to measure one gene expression at particular time with the help of Southern blotting and other techniques. So if we want to measure the activity of 20,500 genes at particular time, we at least required 5 to 10 years for one single study. With the help of DNA microarray, we are able to detect the activity of that 20,500 genes within 1 week. This is the more advantage. This is the drastical advantage of DNA microarray. Okay, so this is the gene expression assays. The main are the short nucleotide, that is the gene chip, and conventional is the cDNA, that is cDNA array developed by the Brown and Boston. Okay, this short oligonucleotide array is developed by the Affymetrix. These are the other are serial analysis gene array, the long nucleotide array, and the fiber optic array. I am going to explain the short oligonucleotide array and complementary cDNA array. Okay, the first is the slide microarray. Second one is the DNA chip microarray. Okay, the each first one is slide. So each slides containing multiple spots, and each spot containing a DNA clone that is generated by PCR machine. Okay, on the other hand, oligonucleotide, we also know as the DNA chip, is not, we are not using the whole sequence of DNA gene, we are using only but very small part of gene. That is the oligo, means very small, and nucleotide is the sequence, very small sequence which we are added to the DNA chip. Okay, now I am going to explain one by one. So first one is the glass slide. It's glass slide, the conventional glass slide invented by the Patrick Brown and his colleagues at Stanford University. So glass slides having multiple tiny spots, that is the array, and each spots having one complementary copy of DNA. Okay, so they attach the DNA with the help of robotic arms to that slide. The slide they are using is made up of the nylon, glass or silicone. Okay, so how they make the chip? It is a very precise method with the help of robots, which is attach metal pins that dip into the DNA solutions and put that DNA solution, which is nothing but the gene sequence, and put that genes on particular spots. So with the help of this video you can easily understand how they do it. So this containing DNA, this is the DNA solution having the gene particular, the robotic arm which is dip into the DNA solution, take the DNA solution and put that DNA solution and attach that DNA onto the glass slides. Okay, it's a very precise method. What is the advantage of glass slide over DNA chip? So the main is lower in the cost. Second one is the prior knowledge of sequence is not necessary, because we are not synthesize any complementary copy, we are directly attach the particular gene to particular spot. On the other hand, in the DNA chip we are synthesize only short oligonucleotide, that is the short sequence of nucleotide. So that's why we know the sequence prior to synthesis of oligonucleotide. Okay, that is the main advantage of glass slides over DNA chip. And the main disadvantage is the density of that DNA genes is very less as compared to gene chip, where we can attach many complementary copy to particular spot. On the other hand, we are using up to the two or three gene copy in particular spots. Okay, second one is the oligonucleotides array. It is the more advanced technique. We are using the we are synthesize the oligo short nucleotide sequence and put that sequence into the DNA chip. Okay, with the help of photolithographic process they are able to make the DNA chip. The advantage of this is the very speed, highly accurate, and the reproducibility. And the major disadvantage is the short sequence. As we know, the sequence is only up to the 10 to the 15 base pair only. Okay, so longer the sequence, longer the sensitivity, shorter the sequence, So this is the normal flow chart of DNA microarray procedure. You are taking sample from the patient, that is experimental sample, and the control can be taken from the same patient or the normal other individual. Suppose the patient is having melanoma of right thigh, the control can be taken from the skin of left thigh, shoulder, axilla, anything from else. Then extracting a mRNA from the patient as well as normal person, add the red color dye, fluorescence dye, to the experimental patient cells, and the red color dye and the green color dye to normal patient or control sample. Then we mix both the samples on the glass slide which is already having DNA. So the DNA gene which is more activated or more complementary will bind to that particular slides and give corresponding result. Suppose if the gene is activated only in the normal person, it will give green color. If the gene is only activated in cancer tissue, it will give red color. If it is activated in both, it will give yellow color. If it is not activated in both normal as well as experimental cell, it will give black color. Okay, it's very simple. Which gene, the gene which is more expressed, give corresponding color. The fluorescence intensity of each spot is proportional to the level of expression. So this is the scanner diagram. Here we can see the green color dot, red color dot, yellow color dot and black color dot. So green color indicate the gene is expressed only in the normal person. Color indicate the gene is expressed in the patient, that is the tumor sample. Yellow color indicate gene is expressed in both, and black color dot indicate gene is not expressed in both. Okay, so if we are taking the mean value, if the mean value is less than 0, it means it is green in color and expressed in the control sample. If it is more than 0, it is expressed in the red color. It means the gene is activated in the tumor cells. If it is equal to 0, it indicate in both the sample and give the yellow color. If it is not expressed, it will give the black color. Again a simple flow chart for those who still not understand the concept of DNA microarray. We are taking DNA from the patient, DNA from the control, extract the mRNA, then add this fluorescence to both the sample. Cyanine 3, that is the green color, to the control, cyanine 5, that is the red color, to the patient. Then mix the both sample and wait for hybridization. The gene which is more complementary to gene which is already present to the DNA slides bind more firmly and give corresponding color. The gene, if gene is expressed in the patient, it will give red color. If it is expressed in the control, it will give green color. If it is expressed in the both, it will give yellow color. Of DNA microarray: collection, isolation, creation, hybridization, microarray scanner, and how to analyze the data. First steps is the sample procedure. We can take either sample, peripheral blood, aspirated fluid, swab sample, lavage, anything which can, any cells which containing DNA can act as a sample. Then the final step is the isolation of mRNA, because if we are extracting the cells, it contains all the RNA, tRNA, mRNA, rRNA, but the RNA of interest is messenger RNA because it's containing code for protein synthesis. So rRNA, we know that the mRNA is having poly A tail on last side. So we are adding a bit which containing poly T tail. So we know the rule of complementary, A always bind with the T. So in this way, the bit having attached mRNA, then we wash the samples. In this way we will get only mRNA and the remaining RNA is washed from the sample. Then adding the fluorescence, the cyanine 3 and cyanine red to corresponding samples. The final step is the hybridization, that is mixing of patient and the control sample to the slide which is already having the DNA. Then the final is the scanner containing the laser, computer and the camera. So this is the animated picture of scanner, the internet. So it having 3 buttons of green, yellow and red. The green, first we scan green, that is the gene which is expressed in the control sample. Then we scan red, the gene which is activated in the patient or tumor cells. Now the final step is the merging of samples, that is merging of scan, that is more important. After merging the image, we will get the picture like this: lots of yellow dot, lots of black, few green and few red dots. So for example, if it is gene number 17 which encode BRCA1 gene, BRCA gene number 17. If we are study the breast cancer, and we will get the same result after many study, we will come to know that the gene number 17, that is BRCA overexpression, is directly proportional to breast tumor. It's very simple. We know all the 20,500 spots, we know the sequence of the gene. With the help of gene expression study, we can know that which gene is particularly activated in which tumor. Okay, this is the final print of scanner copy of DNA microarray and the final analyzing data software. So applications of DNA microarray. What are the main applications of microarray? Obviously in the cancer research, diagnosis, for molecular diagnosis of new cancers, immunology, the genomic response to the bacteria or viral infections. Okay, many virus or bacteria change their genomic interaction, like HIV virus, they change their genomic, resequence themselves after exposing to the certain chemotherapeutic agent. So then non-tumor pathology, like in the neuropathology like Alzheimer, Parkinsonism, epilepsy, schizophrenia, and immunopathological diseases like multiple sclerosis, organ transplantation, where the etiology is not known. Microarray or gene expression is definitely playing a role for future. Now the final and most important in the tumor or cancer. I'm showing the example. Before invention of DNA microarray, diffuse large B-cell lymphoma, some people having the good prognosis and some people having the very bad prognosis. So after molecular study of diffuse large B-cell lymphoma, we came to know there are two types, that is germinal center like and activated B cells like. So activated B cells having the higher mortality as compared to germinal center, because it's showing abnormal gene expression of BCL2, CMYC and MAPK5 kinase. Oncotype DX is the another study which is having 21 genes. Proliferation, which including proliferation gene, estrogen gene and gene for invasion. With the help of Oncotype DX, we came to know that it's a likelihood, what is the likelihood of the recurrence and the magnitude of the chemotherapy, it is whether the chemotherapy is effective or not for particular patient. The next advance is the MemmaPrint, which containing 70 genes, which is finally approved by in 2007 by the US FDA. With the help of that gene, we know whether this particular tumor is metastasized to other organ or not. So this is another example published by the Medical College of Wisconsin. 6-year-old boy Nick suffered from severe inflammatory bowel disease, underwent many surgeries. They mention almost 100 plus surgeries, but still not curable. After whole exomes sequencing, that is 20,500 gene study, they found mutation in XIAP gene, that is the X-linked associated protein gene, which is present on the X chromosome, and this gene is responsible for the immune response or inflammatory response of our body, and the mutation of this gene can lead to severe inflammatory bowel disease, and fortunately this is curable by the bone marrow transplant. So in July 2010, the Nick received the stem cell transplant from the healthy donor and they recover continuously. This is the best example for future of medical practice or genomics. Now another example is FDA approved CYP450 chip, that is for warfarin. For it is a blood thinner agent. The overdose and underdose both are very harmful for the patients. So every individual having the different capacity to metabolize the warfarin. So with the help of CYP455, this enzyme is responsible for metabolism of certain drugs in our body. So we can set the dose of warfarin for every individual. Now This is the example of some gene associated with the cancer. We know that APC is associated with the colon cancer, BCR-A1, AB breast cancer, RB with the retinoblastoma, and all this. And this is the target therapy. Why I am emphasis so much on gene expression? Because we will target the particular therapy to suppress the overexpression of that abnormal gene expression, like trastuzumab for HER2 amplifications. And it is the other benefits of DNA microarray: new gene discovery. Okay, second one is the gene expression profile, as I told in details. Cancer diagnosis and classification, that is for breast cancer, classified breast cancer into the molecular types. Then drug discovery and toxicology, DNA resequencing, sequence of the genome of SARS and HIV viruses. And finally in agriculture and forensic science. Okay, so what is the drawback? Every machine is having advantage and disadvantage. So if I am discussing the advantage, I must include the drawbacks of the microarray. So the basic drawbacks of is lack of standardizations, time in fully automated mode still required 3 to 4 days for report. Then final and the most important is the clinical relevance. It needs larger prospective study to support the use of microarray in routine diagnostic practice, and the most important is the cost. So this is the conclusion. DNA microarray is one of the most effective intervention ever developed, but still it required lots of clinical relevance to support the use of microarray in routine diagnostic practice. Thank you. If you like this video, please subscribe my channel.